Imagine looking into a microscope and seeing in real time a neuron repairing itself after damage. This is the power of fruit fly neuroscience research in the Thompson-Peer Lab at the University of California, Irvine. Welcome to the Thompson-Peer Lab. Today we're following how researchers use fruit flies, formerly called Drosophila melanogaster, to study the brain. We begin in the fly room, where hundreds of different fly lines are stored in vials, organized in trays. Each vial contains flies with unique genetic modifications, labeled with tape to track their traits. Using carbon dioxide, the flies are gently anesthetized. Using a paintbrush, researchers separate the flies by sex and genotype. They look for visible markers, wing shape, eye color, body tone, all clues to the genes inside. Now we head to the microscope room. It starts with a grape juice plate, where larvae are collected using forceps. They're washed in a dish of water, then transferred to a microscope slide. The larvae are placed on a soft agarose pad covered in glycerol and gently pressed under a cover slip. The pressure is just enough to keep them still, while keeping the animal alive and unharmed. The slide is secured onto the microscope stage. The objective lenses rotate into position beneath the specimen. The neuron researchers are interested in expressing the green fluorescent protein, which glows green under blue light. Once the larva is roughly positioned, Vinny switches to laser scanning and computer imaging to visualize the neurons in detail. The neuron appears glowing green on screen, with the cell body at the center and its branching dendrites and axon extending outward. After aligning the neuron precisely, Vinny uses the two-photon laser to make a targeted cut, severing a single dendrite branch. This controlled damage allows researchers to study how neurons respond and repair. That's how fruit flies help unlock the secrets of the nervous system, one tiny step at a time.